How can MRD status be used as a surrogate endpoint in a clinical trial?
The FDA has not yet approved MRD testing as a surrogate endpoint in multiple myeloma. The myeloma community has agreed how to define MRD negativity, and the decision for right now is that a test with a sensitivity of 10 to minus 5, or 1 cell in 100,000, whether it's flow cytometry based or next generation sequencing based, it doesn't matter. But if a patient is negative with a test that has that sensitivity level or better, that patient could be called MRD negative. There are ongoing efforts looking at patients who are treated on clinical trials in the randomized setting, if they reached MRD negativity with the definition I just mentioned, versus if they did not, then they would obviously be viewed as MRD positive, to see whether MRD negativity at a given time point from the start of the study, if that would be predictive of the duration of progression-free follow-up time. If that can be proven, then most likely the FDA will accept MRD as an early read for trials or a surrogate endpoint. And I think this will be very, very important for all patients. It will be important for drug development. It will impact drug companies, and it will also impact academic doctors that are participating in drug development. But the big winners definitely is going to be all patients, because drugs can now be reviewed and approved much, much faster. Instead of having a trial going for 5 or 7 or 8 years, the trial would read out, let's say after only a year, you would know if it's MRD negative, higher or lower in the experimental versus the control arm. And you could get approval, and the drug could actually be available for patients. And I also think, very importantly, for a drug that does not deliver good responses, that drug should be taken off the trial, and patients should not be exposed to the drug, because there is also toxicities related to new drugs. So I think having access to early instruments for readout will be very, very important in the future for all patients.
I think for many years now, the FDA has approved many drugs based on how long it takes for the patient to relapse with a certain therapy. We call that progression-free survival. It may not be the best outcome and may be hard to achieve that in a short period of time, because sometimes it takes you 3 years to see that signal. So the question is that, can we find a better way to say this regimen is very effective? I think the depth of the response, getting rid of more myeloma, is a good thing. So the more myeloma we get rid of, I think the better the outcome will be. So achieving MRD negative result with a new novel combination in a high percentage of patients should indicate that this combination, this regimen, this new treatment is actually worthy of maybe approval or accelerated approval pending confirmatory studies. But I think that should become a good mark. I mean, I think, nobody's going to be cured with partial response. I think if we're going to cure patients, they need to achieve MRD negative state, and MRD negative state that is lasting. So I think it does make sense to consider MRD negativity as a new surrogate marker for activity of a new combination or new drug.